Hello. I appreciate the opportunity to present our data today. I am a developmental biologist in the Department of Molecular Cell and Developmental Biology. And you can see a little bit of the background. Today the project I'm going to highlight is one that's actually a collaboration between four of us, myself, Ben Novich, Mike Jung, of course, and Valerie Arboleda. And the real stars here are the images there. You see these organoids. These are mini brains generated from human pluripotent stem cells, differentiated in a sophisticated way by the Novich lab, that recapitulate many of the hallmarks of different layers in the fetal brain. And so if you section through one of these organoids, you see all the different layers represented that appears almost to mimic exactly a fetal brain at around mid-gestation. And so with these organoids, lots of investigators around the world are doing all kinds of interesting studies in developmental biology to figure out how cells are born. And in my lab, along with Ben's lab, have been thinking about intellectual disability syndromes, which are terrible for families. There's no way to treat these diseases. And they are individually rare diseases, but there are at least 300 monogenic versions of intellectual disability disorders, all of which seem to target proteins involved in epigenetic regulation. And so for some reason, all of these 300 diseases target the same molecular pathways and lead to the same types of phenotypes, namely facial dysmorphia, intellectual disability, short stature, microcephaly. And amongst all of these ID syndromes, at least $50 billion is spent each year just to care for these people, but we're not doing anything currently to actually ameliorate their symptoms. And so in my lab, we use pluripotent stem cells derived from patients with various maladies and do what we call disease in a dish, where we take skin cells from these patients, turn them into pluripotent cells, and then generate cells of interest, such as neurons, and then study how those neurons are defective. And so in collaboration with Novich lab, he took cell lines that we had generated from a disease called Rett syndrome, which is a terrible disorder that affects 1 in 10,000 girls that are born. Boys don't survive because it's an X-linked disorder. And essentially what the Novich lab can do is generate these organoids, and then you see with microelectrodes can actually measure electrical activity and the real physiological activity of neurons in these organoids, to the point that they can actually detect brain waves. So the beta, alpha, delta, theta, gamma rhythms that correspond with awake, sleep, arousal, etc. They can detect these patterns in these organoids that represent mid-gestation brain. So with that, if you look at organoids that are generated, I apologize for the color scheme, if you look at organoids that are generated from these Rett patients, first let's look at the control, is the blue line there. Hopefully you can see the organoids from just wild-type cell lines have these canonical patterns of beta, gamma, theta rhythms. And then if you look at the red, if you make organoids from patients with this intellectual disability syndrome, they have no rhythmic patterns. So they're not undergoing appropriate network activity. On the other hand, if you treat those organoids from the patients with a drug that blocks senescence, and we had a paper to describe this, if you block the senescence in those organoids, now you restore the theta rhythms. So we see this as a very nice screening platform to identify drugs that could restore appropriate neurophysiological function in a model of human brains in vitro. And so next we're going to move this platform to a high throughput approach with multi-electrode arrays. We're doing medicinal chemistry with Mike Jung. We've developed novel molecules that cross the blood-brain barrier to do that. We're looking at other ID syndromes. It seems as though there's at least 300, many of which share the same issue with cellular senescence. And eventually we'd like to create a private entity with partnership. And so with that, I will highlight 3 posters, one of which from Ozzy Miranda that describes this organoid work, another one from Matilde Miranda, who's a grad student in my lab, working on hair follicle developmental biology, and another from Martina Roos, who studies AML, and we've stumbled across a way to treat AML by a novel mechanism. So I thank you for your attention.